In biocompatibility evaluation of medical devices, several in vitro and in vivo tests are performed. Among these tests, cytotoxicity testing is generally considered a simple first-line screening method. Many times the results of the test are negative, meaning the material is not cytotoxic. However, occasionally the results are positive, indicating cytotoxicity of the test material. Despite this, in toxicological analysis, a positive cytotoxicity result is generally not regarded as a major issue, as it may not be truly indicative of in vivo or human toxicity. Here, I aim to break down the cytotoxicity test and provide details about its potential role in evaluating the biocompatibility of medical devices, as well as its limitations. I think this can be useful for professionals in the medical devices industry, and especially those involved in R&D, quality, or regulatory compliance. First, let's see how the test is performed. Cytotoxicity tests are usually conducted using three standard methods. The first one is direct contact. The second method is extract method. And the third method is indirect contact. Now, let's look at direct contact method. In this method, the test material is placed directly onto cultured cells to detect acute toxicity from contact. In the extract method, which is commonly used for cytotoxicity testing of medical devices, the material is incubated in a medium, and the resulting extract is then applied to cells to detect any leachable toxicants. And lastly, in indirect methods, barrier systems, such as agar overlay, are used to prevent physical damage to cells while allowing diffusion of toxic substances from the test material, such as an implant. Similar to other methods, the detection of acute toxicity from contact will be determined. In the next slide, I will discuss major materials-induced toxicity mechanisms that cytotoxicity assay can evaluate. Now, let's discuss these mechanisms in detail. While a variety of cytotoxic outcomes can occur, including apoptosis and oxidative injury, they can be generally grouped into four broad types of cellular damage. Depending on the assay design, these effects can often be detected by in vitro cytotoxicity testing. Here, I briefly review these. The first one is cell death, lysis, which is acute toxicity, where the material disrupts membranes, killing cells outright. Growth inhibition. Cells survive but divide more slowly, indicating sublethal stress. Metabolic dysfunction, impaired mitochondrial activity or enzyme function, measurable with metabolic assays, e.g. MTT, risazurin. And lastly, membrane damage. Loss of membrane integrity with leakage of cell contents, measurable by assays such as LDH release. While cytotoxicity tests do not directly identify the chemical, they detect the effect of a broad range of potential toxicants, such as heavy metals like lead, cadmium, nickel, leaching from alloys or pigments, residual monomers or catalysts from polymers such as formaldehyde, acrylates, plasticizers such as phthalates or stabilizers, degradation, products from sterilization or aging such as peroxides, acids, or organic solvents remaining from manufacturing. Now, let's look at some common chemicals found in medical device materials and the types of toxic effects they can cause, as detected in cytotoxicity tests. Nickel in stainless steel tools, implants, and pigments can damage membranes and trigger allergic-type cell death. Formaldehyde from certain resins and polymers may cause rapid cell lysis and protein cross-linking, as in some dental or molded plastic parts. BPA in polycarbonate and epoxy coatings can disrupt growth and mimic hormones, seen in syringes, tubing, or housings. Acrylates in UV-cured coatings and dental resins may damage membranes and cause inflammation. Phthalates in flexible PVC for tubing, catheters, and blood bags can impair metabolism and slow cell growth. Silver ions in antimicrobial dressings and catheters can disrupt cell function. These findings help identify potential hazards early, but their real-world impact depends on how the material is used in the body. As you saw, the cytotoxicity test is a sensitive first step, allowing early detection of harmful chemicals such as nickel, BPA, acrylates, phthalates, or silver ions in a medical device or its extracts, ensuring only safe materials move forward in development. But is this enough? In reality, although cytotoxicity testing, combined with characterization, may be sufficient for some devices, for many others with complex materials, it is only the beginning. To fully assess a device's biocompatibility, further tests are needed. In the next slide, I will discuss the limitations of the cytotoxicity test. Now, let's see some major limitations of the cytotoxicity test first. It detects only general cell damage. Cytotoxicity tests measure loss of cell viability, membrane integrity, or metabolic activity. They can indicate if a material is harmful to cells overall, but not the specific type of harm. Cannot directly detect genotoxicity. Some substances cause DNA damage at doses that do not cause immediate cell death. No or limited information on mechanism. A positive cytotoxicity result doesn't reveal whether the damage is due to membrane rupture, oxidative stress, metabolic inhibition, or DNA injury. In vitro limitations, cytotoxicity tests are performed under controlled lab conditions that may not reflect real in vivo exposure, e.g., metabolism, immune responses. Short-term focus. These tests capture acute effects, but not long-term effects like chronic inflammation, carcinogenesis, or reproductive toxicity. Now that you have an overview of cytotoxicity testing, you can find further information in ISO 10935, which outlines standardized methods to assess whether a medical device or its materials harm cultured cells. It describes direct contact, indirect contact, and extract approaches, evaluating cell viability, morphology, growth rates, and membrane integrity using assays such as MTT, LDH release, or neutral red uptake. This testing is widely required by regulators, including the FDA and authorities in Europe.